layers or tunics. You have a fibrous tunic, a vascular tunic, a sensory tunic. The fibrous tunic is on the outside. This is what muscle is attached to. And we can divide this fibrous tunic into the sclera and the cornea. So we describe the cornea as being the anterior 1/6 of the eye, and the posterior 5/6 would be the sclera. So outermost layer of the wall, 1, 2, 3 layers. So this posterior 5/6, basically to here, called sclera. You know it as the white of the eye. Provides an attachment point for extrinsic eye muscles. And then the anterior 1/6, it's a clear window. It's called the cornea. Light goes through this clear window. And we end up focusing the image on the fovea. So we want to take all the light energy coming in, bend the light so that all the light rays come to one point, landing on fovea. And that's because the fovea consists of 100% cones, which give us the best acuity and color vision. Okay, so the highest resolution image. When we use our peripheral vision, when the image lands on an area outside of the fovea, out here, over here, it's a little blurry, right? And you know that. When you focus on something, sure, you can see stuff around it. You know there's people and you know there's cabinets, but it's all sort of blurry. If you want to see it really clearly, you focus right at it. The further you get from fovea, the higher the percentage of rods and the lower the percentage of cones. And we'll come back to rods and cones in a minute. So posterior 5/6, white of the eye, sclera. Anterior 1/6, a clear window called the cornea. And then the light goes through the aqueous humor, enters the lens, exits the lens, and the job of the lens is to focus all the light on this pinpoint location. Sclera, cornea. In addition to bending light, the cornea also contains lots of nociceptors. Very, very sensitive. And you know this if you've touched the eye. It's painful. But that's important because we do need to protect the cornea from damage. Right? If it's not a clear window, if it gets damaged, that's going to bend the light as it passes through, and that's going to make the object in your visual field seem very blurry. So from superficial to deep, the next layer or tunic is the uvea. It's the vascular tunic. Within it, two pigmented regions, iris, choroid. Choroid is the posterior 5/6 of the uvea. So that corresponds to the posterior 5/6 of the sclera, right? That first fibrous tunic. So right under the sclera is the choroid. Melanocytes produce the pigment, melanin. As you know, melanin is some version of brown. And here the purpose of melanin is to absorb stray light. In the skin, the purpose of melanin isn't to absorb stray light, it's to prevent UV radiation from reaching nuclei. Here, we don't care about UV radiation, that's not the point. We don't want light bouncing around within the eye. We want to make sure all the light coming in gets focused on one spot on the fovea, and that's it. Think about watching a movie on a big screen TV in your house. If you want to see a very clean, crisp image, good contrast, vivid colors, do you watch that movie in the middle of the day with windows open and lots of sunlight coming in? No, because what does it do to the screen? Washes it out, right? The colors are more dull, the image isn't as crisp. it makes it sort of blurry, right? It interferes with your ability to see the object and the color. Same thing would happen in the eye if you allowed stray light rays to bounce around. It would be like sunlight coming in through the room while you're trying to focus on the big screen TV. So to absorb all that stray light, we have lots of pigment. The iris is also pigmented, and the iris consists of two muscle layers that encircle the pupil, the opening that the light passes through. And one of those layers is innervated by the sympathetic nervous system, and the other one's innervated by parasympathetic. And together they are able to constrict or dilate the pupil. And then a third structure associated with the uvea. It's not a pigmented region, but it's a cluster of smooth muscle called the ciliary body. And this muscle uses these thin little ligaments. It's like denofloss. You might see them in a cow eye dissection. And those attach to the surface of the lens, and they change the shape of the lens. It makes the lens bulge. You only need to do this to focus on a near object. So we don't really need to change the shape of the lens at all when we focus on a far object. So that means sort of naturally, that's what we do. Without any intervention, we can see far up objects pretty clearly. And I'm not talking, a huge distance away. 10, 20 feet, something like that. What's more difficult for us, actually, and you wouldn't think of it this way, but to bring something really close, like being in front of the computer screen or a book. You have to dramatically change the shape of the lens to focus on an object that's that close to you. And that's why it strains our eyes when we focus on near objects, not far objects. Right? It's when we're reading or in front of the computer too long that it strains the eyes. So here's the two layers of muscle. An inner circular layer that functions as a sphincter, and that's what circular smooth muscle does. And an outer radial layer. And these smooth muscle fibers radiate away from the center. Or you could say they run perpendicular to the circular muscle. If you want to constrict the pupil, you contract the circular muscle. That's under parasympathetic control. If you want to dilate, then you contract the radial fibers and pull everything out. And that's controlled by sympathetic. So to fully dilate, we would need to do two things. Turn the dial down on parasympathetic to relax the inner circular layer, and increase sympathetic output to contract the outer radial smooth muscle fibers. And of course, if you wanted to constrict, it would be the opposite. Turn down the dial on sympathetic, relax those guys so that they elongate, and then contract the circular using parasympathetic output. So I mentioned the iris is pigmented. Contains only brown pigment. But how much pigment you have and where it's concentrated within the iris actually accounts for all eye color, not just brown eye color. For example, if you have less melanin and it's concentrated in the posterior part of the iris, then the color that you end up seeing when you look at this person's eye is blue, green, gray. But if they have more melanin and it's more concentrated in the anterior portion of the iris, then when you look at that person's eye, it's going to appear more brown. Light brown, brown, dark brown, almost black. It depends on how much pigment. And this gets to how we see color in general. It's based on the wavelength of light that's reflected off of the object that we're looking at. When you look at a white object, the reason it looks white to you is because all wavelengths from the visible light spectrum, all of them, are being reflected off that object into your eye. You perceive that as white. When you see a black object, the reason it looks black, all wavelengths are being absorbed from that object. None are being reflected into your eye. So everything between white and black refers to certain wavelengths being reflected off the object and not others. So black and white is all and none. All reflected or none reflected. Anything else, only certain wavelengths reflected. And that's what produces red and green and blue and so on. Sensory tunic, right? So as we go from superficial to deep, it was the fibrous tunic, and then the vascular tunic, and now the sensory tunic. And there's two layers to the sensory tunic, an outer pigmented layer and an inner neural layer. So again, when you see pigment, the purpose is to absorb stray light. So we have it in the choroid, and we have it in the retina. The inner neural layer refers to the neurons that provide us with vision. That's the photoreceptors, and the cells that they communicate with as part of the special senses organ. There's actually 3 cells that work together to transduce energy, light energy, into action potentials. So we looked at the wall. Now we're looking at the interior of the eye. Inside the eye, there's a lens. The ciliary body encircles the lens. And here's the dental floss I referred to. Right, that's the ligament that attaches the muscle of the ciliary body to the lens. And the iris is right in front of the lens. And then in front of the iris, between the iris and the cornea, is an open space filled with vitreous humor, or aqueous humor, watery humor. Humor is a very old term for body fluids. We used to refer to all body fluids, including blood, as humors. Behind the lens, in the posterior compartment, a gel-like vitreous humor. So anterior compartment, aqueous humor, very watery. This fluid is constantly coming into the chamber and constantly going out. It circulates, much like synovial fluid circulates in a synovial cavity. Aqueous humor circulates in the anterior compartment. And in part, it's helping to nourish the iris and the cornea. And remember, this whole fibrous layer is avascular. It has to get its nourishment somewhere because it doesn't have its own blood supply. Now, where's this? The sclera getting its nourishment from the vascular tunic just underneath, the choroid. But there's no choroid up here. So we need an alternative, aqueous humor. Posterior compartment, vitreous humor. When you cut open the cow eye, you'll be able to pull this out as one big glob. Gel. What's the purpose? The function of aqueous humor is nourishment of tissue. How about vitreous? Maintain the shape of the eye. Right? If there was nothing in there, it would easily collapse. So back to the sensory tunic, the retina. Here's the outer pigmented layer. It's absorbing stray light. And the inner neural layer. So notice that light actually passes the ganglion cells, the bipolar cells, before it strikes the photoreceptors. And any light that bypasses the photoreceptors is going to hit this wall, this pigmented layer of the retina. And that's going to prevent the light rays from bouncing backwards. We don't want the light bouncing around in the eye or scattering in the eye. Don't worry about the 2 cells with the pink dots, the horizontal cell and the amacrine cell. These guys are involved in very rudimentary processing of visual information. So special senses organs we know are much more complex than general senses receptors. One example of that is we actually start processing, start filtering out information, for example, at the receptor level, in the retina, before the information even gets to the brain. So we'll ignore those cells and just look at the ones that are actually transferring the signal. So the photoreceptors excite the bipolar cells. The bipolar cells excite the ganglion cells. The ganglion cells have really long axons that bundle together, exit the back of the eye as the optic nerve. So it's not the photoreceptors, but instead the ganglion cells that actually take the action potentials from the eye and bring them into the CNS. Two types of photoreceptors, rods and cones. Rods look like baseball bats, the yellow ones. Cones look like squid, the purple ones. You have a mixture of rods and cones in the retina, but there's a definite pattern. The closer you get to the fovea, which is the point at the back of the eye where we want to focus all of the light rays. That's where we want to focus the image of the object we're looking at. There it's 100% cones. As you move away from the fovea, percentage of cones goes down, percentage of rods goes up. You get far enough away, it's just rods, no cones at all. Now the difference is, rods have the benefit of being much more sensitive to light. So if you have very dim light conditions, you can actually see better with the rods than you can the cones. And there's a little trick you can do to see something in dim light. The benefit of the cones, they give you color vision and high resolution. Downside to the rods, no color vision, poor resolution, image is sort of blurry. But here's the trick I was referring to. If you're in a dark room, and I've done this many times, just lying in bed, the smallest amount of light comes in, maybe through a door, maybe through the window, and makes a little shape on the wall, right? Just a little bit of light coming in. If you look right at it, if you focus on it, it kind of goes away. It's harder to see that light or that object that the light is striking, that it's lighting up. But if you look just a little bit away from it, right? Don't focus right on it, but a little bit away so that the image isn't landing on the fovea now. It's landing off to the side where you have more rods than cones. Now it becomes brighter. You can actually see it a little bit better. Has anyone experienced this? I have many times. If there's something that I want to see, I know it's there, I can just barely see it. If I just look a little bit away, boom, it brightens up. I can see it a lot better. Then I go and I focus right on it, it disappears. It's because the light intensity just isn't enough to activate the cones. But it is enough to activate the rods. So the trick to getting the object to land on rods, don't focus on it. Look at something just a little bit to the side of it so that it lands in your peripheral vision, so to speak. It's a problem with the cones. Yeah. There's another spot you need to be familiar with in the back of the eye. So one was the fovea, or fovea centralis. The other one's the optic disc. The optic disc is the area in the back of the eye where the axons of the ganglion cells exit and form the optic nerve. So you can't have photoreceptors here. It's just a big bundle of axons. So literally, there is a blind spot in both visual fields, the whole visual field from your right eye, the whole visual field from your left eye. And if it wasn't for the brain filling in the information, you would literally walk around seeing two big black circles everywhere you looked. Those would be your blind spots. The reason you don't see the blind spots, your brain is taking the information around the blind spot and using it to fill in the blind spot. If you're just looking at a whiteboard, everything's white, right? The blind spot would be a little black circle, but your brain sees white around the blind spot and so fills it in white. to prove that this blind spot exists, take your card. And I have a couple extra if you need one. If you're going to use your left eye, you cover the right. With your left eye, focus on the circle that should be on the right side of the card. The X is going to land in your peripheral vision. Start with it really close, so the dot right in front of your left eye, and then slowly move it away. And right about here, for me, the X disappears. So the reason the X is disappearing is because your brain's taking the information around the X, which is white, and fills it in. And if you want to do the other eye, just flip the card over, cover your left eye. With your right eye, look at the dot, and the X should land in your peripheral vision. Anyone need one?
It's at like a certain spot. Oh.
This is actually just one example
this much distance from me.
of how your eyes, your visual system does not operate like a camera. What you think you see isn't always there, right? Like the blind spot.
So creepy.
That white isn't really there, but your brain fills it in for you. If you think about any optical illusion you've ever seen, like you're looking at a spiraling line, and you know that line isn't moving, right? You could just draw it there, but if you stare at it, it seems like it's moving, like it's spinning in circles, right? Or I had somebody show me this little trick once where he put a card on an overhead projector, and on the screen it was magnified, and he drew 4 little legs on it, something like this. And he said, "What shape do you see?" And we all agreed that we saw a rectangle. It looked like a rectangular table. And he said, "It's not a rectangle." And he pulled the card off the projector and showed us, and it was perfectly square. Okay. And I don't remember exactly how it worked, if there was some shading involved, but it was just another example of optical illusions. Sometimes what we see as a wavy line is really straight, a really straight line. We see it as wavy. It just depends on so many different things. But that's what optical illusions do. They take advantage of the fact that your brain is actually taking a lot of shortcuts. Not everything that we think we are seeing, we're actually seeing. It's the brain filling in, making predictions. So just go to Yahoo or Google, type in optical illusions, and you'll see thousands of them. What's the Exploratorium in San Francisco, I believe, has a whole section on optical illusions. Okay, so going back to that light energy is electromagnetic radiation. So there's a whole spectrum of electromagnetic radiation that includes energy waves from meters, which radio waves are, to nanometers, or less than nanometers, which X-rays are. The visible light spectrum is between X-rays and radio waves, much closer to X-rays. But it's from 400 to 700 nanometers. Electromagnetic radiation in that range, to us, is visible light. We can perceive it, we can see it. Different wavelengths within this range allow us to perceive different colors. So again, the objects that we look at have color based on the wavelength of light that they reflect. Right? And again, the two extremes, white and black. If all wavelengths are reflected, you see white. If none of them are reflected, you see black. If only certain wavelengths are reflected, you see a particular color. And what's interesting is we can see a huge range of colors. I mean, huge. Now, if you're a guy, all we have is what? Green, blue, red, black. But if you're a girl, there's magenta and lavender and coral and peach and all sorts of colors, right? Now, all kidding aside, we all see the same colors. Guys just describe them differently. But we see all of that variation using only 3 different types of cones, referred to as red cones, green cones, and blue cones. And obviously, we see more than just red, green, and blue. So that's going to happen when those cones are stimulated at different intensities and as groups of cones are stimulated by the same light energy. Right? So if you stimulate just red cones, you get one color, just green another, but if you're stimulating a little bit of red and a little bit of green at the same time, you get a completely different color. And we're not going to get into all the details of that, but it is described in the chapter reading. Now, as far as the object itself, we have to focus it onto a pinpoint location in the back of the eye, the fovea. To do that, we have to bend the light. We have to bend it and converge it, right? So it all converges on the same point. The light bends when it goes through the cornea. It bends again when it enters the lens, and bends a third time when it exits the lens. Depending on the distance of the object, the lens can change shape to focus. Depending on the amount of light reflected off that object, we can change the diameter of the pupil to adjust how much light enters the eye and reaches the retina. So again, if it's a far object, we don't have to manipulate the lens so much. We're naturally set up for looking at objects a little further away. If it's really close, we do have to change the shape of the lens to focus. Once the light is focused on the retina, then it's a matter of transducing light energy to action potentials. Phototransduction is pretty complex and described in the chapter reading. The last objective was asking you to be able to identify visual deficits depending on where a particular lesion might be in the visual system. And these are the 3 scenarios. What happens when there's damage to an optic nerve or eye? What happens when there's damage to the optic chiasm? What happens when there's damage to the optic tract or visual cortex? So let's start with optic nerve. Each eye has a left and a right visual field, or a medial and a lateral visual field. Now, just like most sensory information that reaches the cortex, there's crossing over. So our left visual field information. So from the perspective of the subject, right eye, left eye, so the person's looking like this. Blue represents left visual field, right? It's the left visual field of the left eye. It's the left visual field of the right eye. Purple represents the right visual field for the left eye and the right eye. So where does all the right visual field information need to go? Left side of the brain. Left visual field information, right side of the brain. So at some point, the right visual field from the right eye has to cross over to the left. Left visual field from the left eye has to cross over to the right. The crossing over happens in the middle of the optic chiasm. What's going to stay on the same side? Which visual field from the right eye is going to stay on the right side of the brain? The blue, which is the left, right? Because the left visual field of the right eye is already where it needs to be, on the right side of the brain. So it stays ipsilateral. Contralateral, crossing over, is the right visual field information from the right eye. It needs to go over here, and the color coding is helpful. So what if the left optic nerve is cut? What are you going to lose? Are you going to lose all of your left visual field information? All of your right? Some left, some right? What's affected? What are you going to lose? Left and right visual fields from the left eye. What if the optic chiasm is cut right down the center? So all the axons crossing over are damaged. Now what are you losing?
Center line of sight.
You end up with tunnel vision. You're losing the right visual field from the right eye, because that needs to go to the left side. You're losing the left visual field from the left eye, because that needs to go to the right side. Right? And you're cutting all those axons that cross. So you have no right visual field right eye, left visual field left eye. You have tunnel vision. You have tunnel vision. That's what they're showing you. So just the medial visual fields from both eyes, just the center. How about if you cut the left optic tract? You lose right visual field information from both eyes. Right? No right visual field information coming in. I have no idea what that would be like. I don't know if that's something we could even really imagine. How about the overall pathway? How many neurons in this chain to get from photoreceptors to the cerebral cortex? So grand total, 4. Photoreceptor, bipolar cell, ganglion cell, and then the fourth one doesn't have a name, but where are the cell bodies of the fourth order neuron? Thalamus. Right, the ganglion cell gets you all the way to the thalamus. And then from there, fourth neuron, from thalamus to visual cortex. 4 neuron pathway. Takes 3 just to get the information into the CNS. Right, so quick preview of the dissection you're going to do, and then we'll do it.
Welcome to the 3-minute eyeball dissection. Make sure you grab some safety glasses and glove up.
Glove up, dude.
First, I want to show you the tools we'll be using.
That's what she said.
Dissection scissors and a blunt probe. That's all you'll need. You'll notice the eyeball I have here is nice and clean and smooth. Well, that's
Thank
because
you for
I
that.
removed all of this connective tissue, fatty tissue, muscle before I got started here to save us just a little bit of time. If you're doing this dissection in lab, you're probably going to have to remove a little tissue before you begin.
Your eye is going to be 3 times that size with tons of fat.
And let's get started. All right, on the outside of the eye, we need to identify a few surfaces. All of this area out here around the circumference of the eye is the sclera. In front, we have the cornea, and that's actually where we're going to make our first incision. It's going to be right in between the cornea and the sclera. Before we check that out, let's flip the eyeball over to the back, and let's make sure we notice this right here, which is the optic nerve, the remnant of the optic nerve. So make sure you check that out. All right, our first incision is going to be between the cornea and the sclera. We need to gently puncture the eyeball. Now be aware the contents of the eyeball are under pressure. In the front, there is the aqueous humor. In the back of the eye, there is...
It can squirt. Rarely does it happen. I haven't seen it happen. But I would say between a scalpel and scissors probably better. And using the really sharp pointed scissors, just jab it. You know, not like, but slowly, but push really hard. It's super thick stuff. And I wouldn't go right at the junction between cornea and sclera. Just go, if you read the instructions, the dissection instructions in the lab manual, it says go 6 millimeters. So just a little bit away from the cornea, and then cut all the way around it. It would take probably a good 5-10 minutes just to get your eye to look like his by cutting all that fat away. I wouldn't spend too much time doing that. It's not a huge deal. You can see everything you need to see on the inside without trimming away any fat.
It's the vitreous humor. These are under pressure. If you pop this and squeeze the eye, it will spray humor everywhere. Trust me, there's nothing funny about getting covered with humor. All right. Very carefully. Be careful not to stab yourself or anyone around you. There we go. Safely puncture the eyeball. Let me complete this incision around the cornea. All right, we're going to flip this up.
Flip the lid.
Right here attached to the underside of the cornea,
Is the,
this back here,
what's that big round thing?
is the iris
Lens.
muscle. That's what
Lens.
this black structure, this black ring right here is. Right in the middle is the pupil, and right behind that is the lens. Now, if this animal were alive, the lens would be extremely flexible.
And clear.
But now that it's preserved, it's extremely hard, almost
And
like
cloudy.
a marble. Very cool to take a look at.
But cut it open and look
This
at the
is rest
layers.
of the eye. Notice it is full of this jelly-like vitreous humor. We're going to squeeze some of that out before we can continue with our cut.
It's like an egg yolk.
All right, we're going to split the eyeball in half at this point. You want to do this carefully.
Now, fibrous tunic is really hard to cut. You have to have really good scissors, which we
This is
don't
the
have a lot of.
optic nerve there. Be very careful to keep your fingers out of the way. If you're doing this on your own, take your time. You do not need to do it this quickly. I'm a trained professional. On the inside of the eye, you'll notice this first layer right here, this light layer. That is the retina. That's what actually allows you to see. It contains those photoreceptors, the rods.
So it's
and
kind of
cones.
like gray.
You
You'll
need
be
to
able
get
to
rid
peel
of
it
the
out.
retina to see the layer underneath, and that is the choroid. In humans, the choroid is just like this up here. It's black, but in many types of animals,
So you see that gray mass right there? That's the retina. So all the photoreceptors, bipolar cells, ganglion cells, that's all in there. So now all that's left is the pigmented layer. In a human eye, it would all be black. In many animals, cow, cat, you have that kind of abalone shell looking area. That's an additional layer we don't have, and it allows them to see better at night. And it's also why when you shine lights into a cat's eyes, it reflects back. It looks like their eyes are glowing. It's because of that layer. He'll tell you the name of it.
cows, they have an additional reflective element of the choroid called the tapetum lucidum.
Tapetum
that allows them to
lucidum.
see in low light conditions at night. It's also what causes eye shine in many types of animals, including cows. If you look at the retina right behind the optic nerve's right here, and then just inside of here, opposite the optic nerve, you will see the blind spot. That's where the retina attaches to the optic nerve. All right, those are our structures for the eye. I hope you learned something. Join us again for more dissections.